Hi. Yep. Thanks for that awesome introduction, Caitlin. So I'm Gabby Canis, and I'm the microbial ecologist at the reserve. So today we've heard a lot about water quality. And when I think about water quality, I'm reminded of a quote by Aldo Leopold, where he says, "Naturalists live in a world of wounds that only they can see." So when we're looking at these water quality parameters, we need to remember that they're really a way of diagnosing our system so that we can begin the hard work of healing it. So water quality gives us a more focused lens to look at a very complex system. So a lot like how plants are the base of the food webs in land systems, in aquatic systems, we have phytoplankton. And these phytoplankton come in a lot of different shapes and forms, and they're extremely diverse. But one commonality is that they're all grazed on by the word phytoplankton actually comes from the Greek term for plant and drifter. So these tiny drifting plants are actually grazed on by a lot of different things in our system. One thing that grazes on these guys are our zooplankton, which are kind of the animal drifting component of the system. There's also different fish species, which will eat the zooplankton as well as the phytoplankton, and so on, moving up until you have fish eating other fish and us eating the fish because we like to eat our fish. In addition to fish, we also have things like aquatic plants in a healthy estuary, which produce oxygen. And we also have invertebrates like our oysters, which we love to see because they filter our water, filter out those phytoplankton, and also they're pretty tasty too. So when we consider our healthy system, we know, thanks to years and years of research by biologists of seasons past, that there are certain abiotic parameters that can indicate the health of our biotic systems. Pretty much, we have a general understanding of how our biotic system is doing thanks to measuring all of these parameters. And for the eagle-eyed people out there, you might notice that there's one parameter that we've talked about quite a bit that's missing from this list that I have displayed here. And that parameter comes directly from our phytoplankton, and it is chlorophyll A. So chlorophyll A is a bit tricky to explain, but it helps me when I think about what these phytoplankton are. And like Shannon said, they're tiny plants. So they're able to perform the magic of photosynthesis, which is taking carbon dioxide and water in the presence of light to form oxygen and sugars. And this process generally occurs in the chloroplast of the cells, and it's hinged on the phytoplankton's ability to capture light. So they have this really special structure that helps them to capture light. And it kind of is like a giant satellite dish. So light energy in the form of photons will come in, get captured by these pigments, move its way all the way down to the center chlorophyll A, where an electron is waiting to get excited. And in that excitation process, it actually releases light at a very specific wavelength, which we can measure and which we call fluorescence. So we measure chlorophyll A as fluorescence as a proxy for how much phytoplankton we have in our system. But with phytoplankton, like I said earlier, they're extremely diverse. So it's not only important to know how much phytoplankton we have in our system, it's also important to know what types of phytoplankton make up our community. And as we experience changes in our watershed through things like development and industry and the things that come along with that, we can have differences in the input of nutrients through runoff. And we've heard a lot about nutrients, but our nutrients can especially affect our phytoplankton. So just how you fertilize your lawn to get your plants to grow, excess nutrients can get our phytoplankton to grow, sometimes excessively. And this is what we call a bloom. So blooms can take a few different forms. One type of bloom that occurs is when we have a species of algae that grows excessively and forms a scum on the top of the water. So the algae itself may not be harmful, but the scum, if it lingers around for too long, can cause some issues. So the first thing that it can do is block light from reaching the other plants in our system that produce oxygen. potentially causing them to die. And also, if you see in this image, scums typically occur on the surface of the water. So they're preventing additional oxygen from the air getting mixed in with the water. So that also reduces oxygen content in the water. And then these cells aren't living forever. So as they die and decompose, they're actively sucking oxygen out of the system as well. So this kind of forms that trifecta, which can cause that condition that you've heard a few times today, which is called hypoxia or low oxygen conditions. And as we know, there's lots of things that live in our waters, which breathe oxygen, specifically through their gills. And if there's no oxygen, they can die, which we don't like to see. Another form of harmful algal blooms occurs when a specific species grows to excess. And there's a lot of good algae, but there are also some types of phytoplankton that are capable of producing toxins. And these toxins can be very variable. Some can affect animals, some can affect humans. Here in particular, we have concerns for toxins that can bioaccumulate in things like shellfish, because we have a lot of our filtering oysters here that we care about. So things that accumulate in shellfish can be of particular concern to us here. A third type of harmful algal bloom occurs when a specific species starts growing in excess, and because of its physical structure, it can actually cause issues. So some species, like the ones pictured here, have a harmful structure or extensions that can get caught in the gills of fish. And when they're growing to excess, they can clog those gills and actually result in fish kills in that way. So we want plankton in our system, but we don't want too much of it or too little. So in order to establish what a healthy community looks like, we take regular samples and monitor them so we know what a healthy system looks like, and then we can also take action quickly if we see deviations from that healthy system. And we have a few different ways that we monitor phytoplankton here at the reserve. The first you guys have already heard about is our swamp program and also our guano water quality project. And in these two programs, they collect whole water samples from the entire water column. They then preserve those samples and take them back to the lab where our amazing volunteers, specifically Paige Priester, is waiting to count every single cell inside that sample. So that is very tedious, but it gives us a really in-depth look at the community within our system. We also have another program that is led by GTM volunteers and linked with FWC, and that is our red tide monitoring. So we have volunteers that go out once a month and grab water samples, and then test it for some of those water quality parameters that we've heard about. And then they package the samples and send them off to FWC, where they do counts for specific harmful species that they're trying to keep an eye out for. The third type of monitoring that we have here at the reserve is our phytoplankton monitoring program. And this is a program led specifically by volunteers where they go out bi-weekly and take concentrated samples. So you can see up here in the upper right corner, a volunteer, Diane, pulling along a net, and she'll be out there walking around and doing circles for 3 whole minutes to really concentrate that plankton so we can see everything that's in our waterways. And with the phytoplankton monitoring network, they're also looking for specific potentially harmful species in those samples. So a common thread that you might have caught on to with all of these monitoring programs is that they're really made possible by the fact that GTM has such involved volunteers and such an amazing group of citizen scientists who help us do research. So today, I'm going to be focusing on some of the findings primarily from our plankton monitoring network, focused at 2 of the sites in Guana Lake and Guana River. So like I mentioned, the plankton monitoring network focuses specifically on some harmful species. And today, I'm going to be talking about 2 of those, which is Chaetoceros and Pseudonychia. So Chaetoceros is one of those physical damage species that I kind of teased a little bit before. And if you look at these pictures, you can see that the cells of this phytoplankton actually have these interesting spines that extend from the main body of the cell. And these spines are what can get caught in the gills of fish when they grow to excess. And we've actually had quite a few incidences of Chaetoceros in our system. So just to orient you, this graph shows all of the incidences or observations of Chaetoceros that we've seen since the Plankton Monitoring Network began in 2012. And we have three different categories of these incidences. We have when they're present, when they're elevated, and when they're abundant. So you can see we've had quite a few times when Chaetoceros has been abundant in both Guana Lake and in Guana River. And specifically, we were able to link one of these events in 2017 where we saw high abundances in our data, and then we saw a resulting die-off of fish in our area. So this was an example of how this data allowed us to pinpoint a cause of a fish die-off, a biological implication that we were pretty concerned about. Another species that we keep a major eye out for here, like I mentioned earlier, is Pseudonychia. So Pseudonychia is a diatom that actually produces a toxin called domoic acid. And this is a neurotoxin which can accumulate inside of oysters. And when you eat that oyster, it can actually cause short-term memory loss. So for people like me who already struggle with memory issues, this can be a really big thing. Like you need to be able to remember things. So we monitor our Pseudonychia because we care about our oysters and we have an active oyster fishery in the Guana River. So luckily, although we have seen Pseudonychia present in our waterways, we haven't really seen too many incidences of blooms of this species. The last recorded one was back in 2014, and it occurred both in the lake and in the river. And we haven't really seen elevated levels of this particular species since then. But if we had, we would want to then take steps to notify the people who are utilizing the fishery in the area to let them know that this is a potential toxin. So maybe they stop collecting for a while until we see levels go back down. We also monitor for common species. This particular species is Cossinodiscus. So it's one that's very bioavailable. So a lot of those grazers that I was talking about, they're munching on this stuff. So we want to make sure that it's present in our system. And as you can see by this basically solid block, it is very present in our system. But occasionally we do get blooms of this that we want to keep an eye on in both the lake and in the river. In addition to monitoring specific species, we can also look at the record of the community as a whole and start to make some inferences and kind of play detective to see the story of our system. So that's what this graph here is showing. So in blue, we have the overall density in cells per milliliter of phytoplankton in the lake. And then in orange, we have the overall density in the river. So the first thing that we see is that our blue line is much higher than our orange line. So that matches up with a lot of the data that Olivia shared that shows that we have very high levels of phytoplankton in our lake. The next thing that kind of stood out to me were these two peaks that we see here. So when we see extremely high peaks of phytoplankton, that's when we want to start digging in because we could potentially see impacts of blooms. So what was happening during these two peaks? Well, thanks to Olivia and all of the data she collects, we can start to tell a story there. And what we see is that during this first peak, we actually had some construction going on at the dam. So that means that there was no water flowing through the dam at that time. And then as soon as it was reopened, we saw that level go down. And then again, during the second peak, the dam was under construction again. So we saw that closing. But essentially what this is telling us is that the dam is reducing that residence time and really helping us keep the populations of phytoplankton to more healthy levels. So main takeaways from this are citizen science-led monitoring can be a really effective tool for water quality. We also know that different types of phytoplankton monitoring strategies have different resolutions. And we know that consistent monitoring can help us identify patterns in our system. In the future, we hope to further link our phytoplankton patterns to water quality, increase data access, and use phytoplankton patterns to inform management strategies. So thank you so much for letting me present. And again, just to reiterate, the main goal of this research is to identify patterns that suggest wounds in our system. And once we know about these, we can really begin the real work of healing those wounds. So thank you.
Thank you so much, Gabby, for giving that presentation and getting everyone excited about phytoplankton. We do have a question in the chat box, and I believe that the question popped in when you were talking about pseudonychia. Do you know what might have been causing the spikes in pseudonychia?
That's a great question. So we don't have quite as extensive water quality from back in 2014, but we are beginning to try to link our water quality patterns to our patterns in phytoplankton at some of our other sites. So hopefully I can fill in those links in the future, but that's part of the project that we're still developing out, is really trying to understand why we see certain species when we do, and identifying the patterns of blooms.
Great, thank you so much. There's some really great comments on how you illustrated plankton, bringing it into the entire estuarine ecosystem. So thank you very much. Do we have any additional questions for Gabby this morning, either in the chat box or with your raised hands? Got one that just came in the chat box. Have you had a chance to look at other places that volunteers have collected data?
Great question. So we actually have data at not only the 2 sites that I mentioned, but we have additional data at the 4 water quality sites, swamp sites that Shannon mentioned. And we're just beginning looking at those sites as well. And there's a 3rd PMN site that is also pretty important. So we're really just starting to delve into this data. So stay tuned, because maybe next State of the Reserve we'll have a lot more data to share about this story.
Great, thank you so much, Gabby.